Hi, I'm Aurel Enriquez, and this video contains our discussion on Chapter 16 of Mahon's textbook of Diagnostic Microbiology, which talks about the clinically significant aerobic gram-positive bacilli. Let's start with the non-spore forming, non-branching, catalase-positive bacilli, which would include Corynebacterium, Rorthia species, and Listeria monocytogenes. The genus Corynebacterium is a large, diverse group of bacteria that includes both animal and human pathogens, as well as saprophytes and plant pathogens. All right, so ang Corynebacteria daw po, it has the capability to infect humans, animals, and sometimes even plants. And some of them could even be seen as free living bacteria, diba? So marami silang iba't ibang habitats where we could find them and they're capable of infecting different species. All right. So most of the species are found as normal flora on the skin and mucous membranes of humans and animals. Corynebacteria can be divided into non-lipophilic and lipophilic species. So when we say lipophilic, it has the ability to dissolve or be dissolved or absorb lipids. All right. So ayan po. Again, when we say lipophilic, they have the capability to either dissolve, be dissolved, or absorb lipids. Okay? So, ayan po. Lipophilic Corynebacteria are often considered as fastidious and grow slowly on standard routine culture media. Medyo hindi daw po madaling patubuin ang mga organisms na to dun sa mga routine culture media lang na ginagamit natin. So cultures must often be incubated for at least 48 hours before growth is detected. So just a review, ilang hours natin usually ini-incubate yung mga samples natin in the laboratory? 18 to 24 hours, diba? But for Corynebacterium species, we might need to extend the incubation for up to 48 hours. However, growth is enhanced if lipids are introduced into the culture media. Okay, so doon na po papasok yung pagiging lipophilic nila, di ba? They would like to be supplied with lipids. All right, so again, they are fastidious and slow growing, but growth is enhanced if lipids are introduced into the culture media. On gram stain, Corynebacteria are slightly curved gram-positive rods with non-parallel sides. Hindi daw po pantay yung sides nila. So they also have slightly wider ends. Mas malapad daw po yung dulo ng bacilli, producing the described club shape or form. Okay, so ayan po, no, when we say form, it has this club shape appearance where the ends are wider in comparison to the rest of the body or length of the bacilli. Right. Another term that we need to be familiar with is the term diphtheroid. So when we say diphtheroid, this means diphtheria like. Okay, so this is sometimes used in reference to this gram staining morphology. If, for example, we're looking at a different bacterial genera, pero medyo mukha silang coriniform, no? So they may be described as diphtheroid, diphtheria like. Let's now talk about specific species, starting off with Corynebacterium diphtheriae. Humans are the only natural host for Corynebacterium diphtheriae. The bacteria are carried in the upper respiratory tract and spread by droplet inhalation or hand to mouth contact. So, an effective toxoid vaccine is available and it is part of the trivalent diphtheria, tetanus, and pertussis vaccine. Okay, ulitin po natin, mayroon naman pong bakuna laban sa diphtheria. So it is included in the trivalent DPT vaccine. Kapag sinabi po natin na trivalent, this vaccine has been formulated to be effective against three different infections. And again, we have diphtheria, tetanus, and pertussis. Well, not really, I don't think effective against is the correct term, but rather it could help us, no, it help our immune system and be protected against those three diseases. Okay, so ayun po. When you get to your microbiology 2, virology classes, no, tsaka nyo po mapag-uusapan yung medyo mas in-depth na overview on vaccine production. Diphtheria toxin is the major virulence factor associated with Corynebacterium diphtheriae. This toxin is produced by strains of Corynebacterium diphtheriae infected with the lysogenic bacteriophage, which carries the tox gene. Okay, ulitin po natin. Ang primary virulence factor ng Corynebacterium diphtheriae is the diphtheria toxin, but they won't be able to produce the diphtheria toxin if they have not yet been infected by a lysogenic bacteriophage. Now, if you would remember from our chapter 1 discussion, a bacteriophage is a virus that would specifically infect bacteria. So usually itong mga bacteriophage na po na ito, sila din yung nagdadala ng plasmids, di ba? And plasmids, they usually code for toxin production or antimicrobial resistance or other virulence factors. So again, in this case, no, not all not all strains or not all isolates of Corynebacterium diphtheriae can produce the diphtheria toxin unless they've been infected by the lysogenic bacteriophage that would carry the tox gene. So basically, it's the tox gene that's going to encode for the production of the diphtheria toxin. So non toxigenic strains can be converted to tox positive by infection with the appropriate bacteriophage. Diphtheria toxin is a protein that is composed of two fragments, fragment A and fragment B. The toxicity is caused by the ability of the diphtheria toxin to block protein synthesis in eukaryotic cells. Okay, ulitin po natin. The diphtheria toxin can block or somehow hinder protein synthesis for eukaryotic cells. So fragment A is responsible for cytotoxicity, and fragment B binds to the receptors of human cells and mediate the entry of fragment A into the cytoplasm. Okay, ulitin po natin, no? Ano yung dalawang fragments ng diphtheria toxin? A and B. Ano ang purpose nila? Yung B, magbabind muna siya dun sa receptor ng host cell. Pag nakapag-bind na siya sa receptor ng host cell, imagining open a door. Parang pagbubuksan niya ng pinto para makapasok ang fragment A, and then fragment A starts doing all the other cytotoxic processes. Okay? So, ayun po. On reaching the cytoplasm, fragment A would disrupt protein synthesis. Okay. So when this happens, this could cause tissue necrosis and exudate formation. All right. Tissue necrosis. So there are parts of the infected tissue na mamamatay na. But there's also exudate formation. So tatakpan niya yon, yung nasiraan niya na tissues, tatakpan niya ng exudates, leading to what is known as a pseudomembrane. All right, pseudomembrane formation. Corynebacterium diphtheriae causes two different forms of disease in humans, respiratory and cutaneous diphtheria. Let's first look at respiratory diphtheria. The incubation period averages from 2 to 5 days, and the illness begins gradually and is characterized by low grade fever, malaise, mild sore throat. And the most common site of infections is the tonsils or pharynx, as we can see right here in this picture. So the organisms rapidly multiply on the epithelial cells, and the toxigenic strains of Corynebacterium diphtheriae produce toxin locally at the specific site where they're currently infecting, causing tissue necrosis and exudate formation, triggering an inflammatory reaction. So this combination of cell necrosis and exudate formation would lead to the production of a tough gray-white pseudomembrane which attaches to the tissues. So it may appear on the tonsils and then spread downward into the larynx and trachea. So as we can see right here on the photo to the left, no, yung let me use my pointer. I know this thing that we're seeing right here, that's the pseudomembrane. Notice that it is attached onto one side of the patient's tonsil. Now, the problem with this one is as it spreads downwards, we can't actually gauge how huge the pseudomembrane is. Hindi natin makita kasi nasa looban na nga, di ba? Which is why we need to be very careful when collecting samples in cases of respiratory diphtheria because sometimes we may accidentally dislodge the pseudomembrane and it could lead to a choking hazard. Okay? So ayan po, that's for respiratory diphtheria. Next, ayan po, is cutaneous diphtheria. So cutaneous diphtheria, in some references, this has been referred to as the Welt sore or the Barcoo rot because it almost looks like the patient's flesh is rotting away. All right, so cutaneous diphtheria consists of non-healing ulcers with a dirty gray membrane. So it's the same. There's necrosis and an exudate formation. So parehas lang naman ng respiratory diphtheria, nasa ibang location lang. Okay, so that's for cutaneous diphtheria. Corynebacterium diphtheriae is highly pleomorphic. Again, just a review. When we say pleomorphic, it means that they could vary in appearance, di ba po? Although they are classified as gram-positive rods or curved gram-positive rods, they may appear different from time to time, no? So, but if we were to perform gram staining, no, the usual observation is that they are gram-positive bacilli that appear in palisades. So when we say palisades, they're kind of like cells that are aligned in rows. Okay? So parang magkakatabi at magkakasunod po sila. Parang nakahilera sila as we can see right here in this example. So that's what it looks like if the organism is in a palisade. Or we could also observe them as individual cells lying at sharp angles that would form a V or L formation as we can see right here. So ito yung itsura ng palisade. Tapos ito naman po yung itsura ng V or L formation. Notice that some of them are kind of like joined together at one side and ayun nga po, parang may angle tayo na ma-observe. So again, that's the V or L formation of Corynebacterium diphtheriae on gram staining. Okay? Um, these organisms often stain irregularly, especially when stained with methylene blue, giving them a beaded appearance, as we can see right here. So, ayun po, no, pag methylene blue daw po ang ginamit natin, irregular yung staining pattern, giving them a beaded appearance. Up next, ayan, we could also make use of metachromatic stains. Okay, so metachromatic areas of the cell which stain more intensely than other parts are called the Babes Ernst granules. All right, so ulitin po natin yon. We're talking about granules that are present inside of the cell. For Corynebacterium diphtheriae, these are referred to as the Babes Ernst granules. But we could also refer to them as volutin granules or metachromatic granules. Okay, so we're talking about the same structure but it has three different names. Okay, so medyo madami po yung tatandaan nyo para dito. Again, the granules of Corynebacterium diphtheriae are more commonly referred to as the Babes Ernst granules, but they could also be referred to as metachromatic granules or volutin granules. So they represent the accumulation of polymerized phosphates, all right? And these polymerized phosphates, no, they contain polymerized inorganic phosphate, which is an energy rich compound. Okay, this is an energy rich compound that acts as a reserve or store of energy and of phosphate. So basically, metachromatic granules or the Babes Earth granules are nutrient reserves. Okay, they're made up of nutrient or they are nutrient reserves. Okay, and what are they made up of? Polymerized phosphates. And so we could visualize the presence of these Babes Earnst granules if we make use of the Albert stain. All right. So for the Albert stain, this makes use of malachite green. And in this photo right here, yung bacterial cell, it has been stained green. Diba? But we're also seeing areas that look like brown circles or brown dots, diba? So yung mga brown areas po na yun, those are where the Babes Earth's granules are located. Corynebacterium diphtheriae is a facultative anaerobe, but it grows best under aerobic conditions, and it has an optimal growth temperature of 37 degrees Celsius, although multiplication would still occur within the range of 15 to 40 degrees Celsius. Okay, ulitin po natin yon. Ang Corynebacterium diphtheriae ay facultative anaerobe, and its optimum growth temperature is at 37 degrees Celsius. Pero ano ang range ng temperature na kaya pa rin niyang mag-grow? 15 degrees hanggang 40 degrees Celsius. Now, on BAP or SBA, the organism can have a very small zone of beta hemolysis. Corynebacterium diphtheria is or better growth, sorry. Better growth is usually obtained on a media containing blood or serum, such as the Loeffler or pie agars. Okay, ulitin po natin yun, no? We could grow this organism on BAP or on CAP, but again, no, there are better culture media out there, such as the Loeffler serum or pie agar. All right. So the characteristic morphology, no, is demonstrated well when the organisms are grown on the low-floor medium. Okay? So kapag low-floor medium po ang ginamit natin or low-floor's agar, dito natin mas ma-observe yung production ng metachromatic granules. All right? Let's repeat that. On the previous slide, we talked about the capability of C. diphtheriae to produce metachromatic granules or the Babes Ernst granules. Now, if we use the Loeffler's agar, it has the capability to further induce the production of metachromatic granules for better visualization. Okay, so that's for the Loeffler's medium or pi agar. Next is the cysteine tellurite blood agar or the CTBA. So the cysteine tellurite blood agar, this is a modification of the Tinsdale medium, which contains sheep RBCs, bovine serum, cysteine, and potassium tellurite. Okay, so CTBA is both a selective medium and a differential medium. Okay, so ulitin natin, no, or review lang po. When we say it's selective, it prevents the growth of a certain group of bacteria, but it allows the growth of a specific group of bacteria. Di ba po? It selects which bacteria is going to grow. Next, kapag sinabi naman po natin na differential, it's going to show us different appearances or colony morphologies of bacteria depending on their metabolysis or reaction to certain components or substrates within the culture media. All right. So again, it's saying right here that the cysteine tellurite blood agar is both selective and differential. So the potassium tellurite inhibits many non-coriniform bacteria. Okay, ulitin po natin yung component ng CTBA na potassium tellurite, it's going to prevent the growth of non-coriniform bacteria. So when grown on CTBA, Corynebacteria could form black or brownish colonies from the reduction of tellurite. As you can see right here in these photos. All right? So kapag po ganito yung itsura nung colonies na na-observe nyo and you were using the cysteine tellurite blood agar, then this is a good indication that your isolate is probably Corynebacterium diphtheriae. On this slide, let's look at some differential tests that we could do to differentiate C. diphtheriae from other Corynebacteria. First is the urease test. It detects urease production or the ability to hydrolyze urea. Okay? So alamin muna natin ano ang components ng or anong culture media pala ang ginagamit dito. No? So for this one, we make use of the Christensen urea agar. All right? Let's repeat that. For the urease test, you could make use of the Christensen urea medium. Okay? So, the Christensen urea medium contains urea, diba? As the name implies. But here's what's going to happen. It also has a pH indicator. So for this one, hydrolysis of urea leads to the production of ammonia and carbon dioxide, leading to an alkaline environment. Um, let's rewind to that time where we talked about um sugar fermentation or carbohydrate fermentation. Diba? Review lang ah. In carbohydrate fermentation, what are the end products? Acids, 'di ba? So acids, no, they lead to the production of an acidic environment. And usually, gagamitan natin ng phenol red para ma-observe natin yung acid production. So when that happens, phenol red is eventually converted and it becomes yellow, 'di ba? But babalik rin natin yung spectrum dito ngayon. If the organism was able to hydrolyze the urea that was present in the culture media, it would produce an alkaline environment. So, kabaliktaran. So, this alkaline environment, hindi rin natin to makikita. Okay? An alkaline environment is colorless, which is why we still need the help of phenol red. So, when the alkaline environment or alkaline byproducts reacts with phenol red, it would lead to a positive result of a color change from ito po parang yellow-orange na culture media to a pink or magenta color formation. Okay, so again, I hope you were able to understand that, no? The urease test, it's supposed to detect urease production. So basically, the Christensen urea agar, it has urea. If the organism produces urease, it will hydrolyze the urea present in the culture media. When urea is hydrolyzed, it produces ammonia. When ammonia is present, the environment would become alkaline. So the alkaline environment would react to the phenol red. Ang phenol red po, it would change its color and it would become pink or magenta, as we can see right here in these examples. So there are many other bacteria out there na magpa-positive dito. So you need to remember, Corynebacterium diphtheriae would test negative for the urease production. Okay. Up next, all right, up next we have the gelatin hydrolysis test. So this detects the production of extracellular proteolytic enzymes, specifically gelatinase. Okay, so sinasabi po dito that there are bacteria out there that could produce the enzyme gelatinase. So for this one, we could use a gelatin stab wherein the agar is replaced with gelatin as a solidifying agent. All right. So if you would remember from our laboratory classes, 'di ba? Sa lab classes natin, we measure and then um we kind of like mix together this solution of media that's supposed to become a solid culture media, 'di ba? But again, remember that that formulation is made up of agar. So para magawa natin ang gelatinase test, yung media na gagamitin natin, dapat yung agar component ay pinalitan ng gelatin. Okay? So they may look very similar but they still have differences in terms of structure. So the for this one to perform the gelatinase test, you need a culture media wherein the agar has been replaced with gelatin as a solidifying agent. So, if the organism produces the enzyme gelatinase, it's capable of hydrolyzing gelatin. So, a positive result is partial or total liquefaction on the inoculated tube. As we can see right here, so, pag sinabi nating liquefaction, parang nag-melt or natunaw po yung culture media na ginamit natin, 'di ba? So, in this example, tube A is positive for gelatinase and tube B is negative for gelatinase production. Kasi sa tube B, yung organism, hindi siya nag-produce ng gelatinase, so it wasn't able to break down the structure of the culture media. So again, there are many other bacteria out there that is gelatinase positive, but ang Corynebacterium diphtheriae, they are negative. Now, the identification of an isolate as Corynebacterium diphtheriae does not mean that the patient has diphtheria, because if you would remember from our discussion earlier, not all isolates of Corynebacterium diphtheriae is toxigenic. All right? Diba? Hindi naman lahat sila ay na-infect ng lysogenic bacteriophage. So, hindi lahat sila nabigyan ng tox gene. At kapag wala silang tox gene, hindi sila makakapag-produce ng diphtheria toxin. And ano nga yung diphtheria toxin? It's the primary virulence factor of C. diphtheriae, 'di ba? So pag walang tox gene, hindi kayang mag-produce ng diphtheria toxin. Pag hindi kayang mag-produce ng diphtheria toxin, hindi kayang maging pathogenic. Okay? So we have right here the ELEC test. All right? So the diagnosis of diphtheria is established by also demonstrating if the isolate produces the diphtheria toxin. Okay? Because again, ulitin natin, not all isolates of Corynebacterium diphtheriae produces the diphtheria toxin. All right? So we have right here the ELEC test. The ELEC test, for this one, each organism is streaked in a single straight line that is parallel to each other and in a minimum of 10 millimeters apart. Okay. After that, a filter paper strip that has been impregnated with diphtheria antitoxin is laid along the center. Okay, so tignan po natin itong example na to. So itong filter paper strip na to, meron siyang antitoxin against the diphtheria toxin. Okay? So, yan po. Um, before we put down the paper, dapat na-streak na muna natin yung mga bacterial isolates natin. Okay? So, again, a filter paper that has been impregnated with the diphtheria antitoxin is laid along the center of the plate on a line that would create a right angle to the inoculum lines. Okay, so dapat parang pa-cross siya. So kung kumbaga parang pa-horizontal nyo po in-streak yung mga bacterial isolates nyo, dapat yung paglatag nyo po ng filter paper strip is pa-vertical. All right. So it would create these kind of like specific position and angles. So after that, no, this would create right angles to the inoculum lines of control and unknown organisms. So gagamit din po tayo ng known and known toxigenic and or sorry, naguluhan ako dun. Dapat po, no, meron din kayong control that is known as toxigenic, meaning kapag sinabi mong known toxigenic, magpa-positive siya palagi, 'di ba? And then you have what is seen right here as a known non-toxigenic. So pag sinabi nating known non-toxigenic, alam mo na palagi ang result niya ay negative. Okay? And then in the middle of that, dun mo po i-streak yung mga unknowns mo or yung mga patient's isolates. All right? So the plate is incubated at 35 degrees Celsius and then it is further examined after 18, 24, and until 48 hours. So a white precipitin line should start about 4 to 5 millimeters from the filter paper strip and are at an angle of 45 degrees to the line of growth. As we can see right here in this example. So if this is our dish and ito yung filter paper strip na may antitoxin, yung mukhang wala silang hindi, sige po. Let's sorry. Let's try to interpret the results that they have right here. So again, if this is our petri dish or agar culture at andito yung filter paper strip, i-interpret natin, no, ang choices natin ay toxigenic or non-toxigenic. So organism 1, is this toxigenic or non-toxigenic? It's toxigenic, 'di ba? Because a positive result is indicated by a precipitin line such as these ones right here. So yung mga guhit dito sa gilid na parang pag ganun, no, that's an indication na nag-produce ng toxin ang organism na to. Next po, isolate number two toxigenic or non-toxigenic? It's non-toxigenic, 'di ba? Kasi wala eh. Walang mga lines na nag-form dito sa gilid. Next, number 3, also non-toxigenic. Number 4, toxigenic. Because again, precipitin lines were formed right here and right here. Okay, so that's for the ELIC test. A Schick test is a skin test used to determine the susceptibility of an individual to diphtheria. The procedure involves intradermal introduction of a small amount of diphtheria toxin into the arm of the individual who is suspected of having diphtheria. A positive result is indicated by redness and swelling on the site of injection, as we can see right here in this illustration. Rhinobacterium diphtheriae also has 3 different biotypes. So when we say biotype, this is just to simplify. This is the same species that's capable of producing a different characteristic. In this case, all of these are all Corynebacterium diphtheriae. Okay, it's the same species, but with regards to its biotype, it's capable of producing different colony morphologies. Okay, so again, just to simplify, no, biotypes sometimes could be the same species capable of producing different colony morphologies. So again, Corynebacterium diphtheriae has 3 different biotypes. First is intermedius. Intermedius is capable of producing small, flat dry and grayish black colonies, as we can see right here. The problem with this one is it's unremarkable. No, there may be other bacterial organisms capable of producing the same appearance. Next biotype is mitis. All right, so the Corynebacterium diphtheriae mitis biotype is capable of producing convex fried egg appearance colonies, as we can see right here. So they are also small and black. Okay, so bakit siya tinawag na fried egg? Okay, sa paligid, parang may white-ish na areas, tas nasa gitna yung black. So supposedly the black area is the yolk. I don't I don't really understand the people who saw this for the first time and said, "Ah, yes, egg." It to me it doesn't look like an egg, but okay, sure, I guess. But that's for the Mitis biotype. Okay. Again, it has been described to have a fried egg appearance just because it looks like it has a yolk looking thing in the middle. All right. Up next is the Gravis biotype. So they're capable of producing large daisy head colonies, as we can see right here. Now, this one is accurate, diba? I-compare mo sa Mitis at fried egg. Is your fried egg rotten? Nabubulok na ba ito? It's a weird looking fried egg. But anyway, oh, anyway, oh my God. The Gravis biotype, mas accurate naman po yung description, di ba? It has a daisy head appearance and the colonies are much visibly larger. Okay, so again, these are the three biotypes of Corynebacterium diphtheriae. It's the same species, just capable of producing three different colony morphologies. Again, intermedius, they are small, flat, and grayish. Mitis, they are black, convex, and fried egg in appearance. And next is Gravis. They are large and they have a daisy head appearance. Enumerated here are other Corynebacteria. So for this one, yung mga mahalagang points na lang po yung tatandaan natin kasi hindi rin naman po sila mga common isolates. So first up is Corynebacterium amicolatum. This is also a normal flora of the skin. It could cause prosthetic joint infections, peritonitis, and bloodstream infections. And take note of the list of the different antimicrobials that it could develop resistance to. Next is Corynebacterium jacium. So, Corynebacterium jacium is also a normal flora of the skin, but it's a common diphtheroid that could cause prosthetic heart valve endocarditis. Okay, so wag pong kakalimutan yon. The disease associated with Corynebacterium jacium is prosthetic heart valve endocarditis. Now, this is also urease negative. No, next is Corynebacterium pseudodiphtheriticum. This one is a normal flora of the nasopharynx, and it has been observed to cause ventilator associated pneumonia, okay, which is secondary to COVID-19. So if, for example, a person has had COVID-19 and they have been admitted to the hospital, eventually it was observed that they also develop pneumonia, which this time is caused by Corynebacterium pseudodiphtheriticum. Notice that this one is urease positive. Next is Corynebacterium pseudotuberculosis. Now, this one is a veterinary pathogen. This is more associated with animals. Now, so, human infections usually result from contact with sheep. Okay, so those sheep, they're the ones who's carrying the bacteria. This leads to granulomatous lymphadenitis, and it's also capable of producing brown halos, brown halos on the colonies if we grow them on the CTBA agar. All right, so medyo magkahawig daw po, no, or very similar, no? Very similar ang appearance ng Corynebacterium pseudotuberculosis with Corynebacterium diphtheriae kung ang gagamitin nyo ay CTBA. Now, the differentiation that we could look into is urease. Kasi diba, Corynebacterium diphtheriae is urease negative. All right? But pseudotuberculosis is urease positive. All right? So, yun po yung, yun yung importance ng mga differential test na dinidiscuss natin. Next is Corynebacterium striatum. This is a normal flora of both the skin and nasopharynx. And do take note that this is only isolated as a contaminant most of the time. Next is Corynebacterium ulcerans. Now, this one, its transmission is primarily through contact with animals or pwede rin naman pong ingestion of unpasteurized animal products, which would lead to a diphtheria-like illness. But again, um, if we were to use the cysteine tellurite blood agar, similar na naman po ulit ang appearance nito with Corynebacterium ulcerans. Alright? But or similar ang appearance ito with Corynebacterium diphtheriae. But again, if we were to look at urease tests, no, the urease of ulcerans is positive and Corynebacterium diphtheriae is negative. Next is the Rothia species. So this actually belongs to the family Micrococcaceae. First up is Rothia mucilaginosa. So listed here are the different diseases associated with this bacteria. The problem with this one is it's quite difficult to memorize all of the diseases associated with them. Kasi 'di ba, kung mapapansin nyo, ang dami na ng mga napag-usapan nating bacteria ang kayang magdulot ng endocarditis, ang dami ng mga bacteria na pag-usapan natin na kayang magdulot ng pneumonia. So, Rothium mucilaginosa, let's look at its gram stain and culture. It has been described as a cocoid. Okay, so doon natin siya ma-differentiate, no? Parang cocoid yung appearance niya. Hindi siya yung typical na bacilli in shape. Up next, we also have Rerthia dentocariosa, which is a normal flora of the oropharynx. So it's quite easy to remember, di ba? Dental, all right? So yung dentocariosa nasa oropharynx. So this could resemble coriniform bacilli. On gram stain daw po, maaaring yung appearance niya is katulad ng mga coriniform bacilli that are slightly curved, gram positive with uneven ends. All right? Up next is Rerthia area. Okay. Sounds like a brain rot name. Rerthia area. Okay, so this was first isolated and discovered in 2004. So medyo recent lang po ang discovery ng Rerthia area. So this was isolated from the air of the Russian Space Station Laboratory. All right. So let me just read off from what I've searched dito sa National Library of Medicine or NIH, no, National Center for Biotechnology and Information. They've described Rotha area as a mimicker of the Nocardia species. Ang dami daw pong similar characteristics ng Rothia era and ng Nocardia species. We'll talk more about Nocardia later. So the description for Rothia area is that this is a gram-positive filamentous bacteria. So filamentous po din po kasi ang Nocardia. Next, it rarely causes respiratory infections, but it should be differentiated with Nocardia species. And the discrimination between Nocardia species and Rothia area needs further analysis using the Malditoff. Okay, so medyo molecular na po yung kakailanganin natin na mga tests for us to be able to really tell it apart in comparison to Nocardia species. Kasi nga po, sobrang similar nila. Alright? So, ayun po, no, on culture, they appear to be white, smooth, which would eventually become dry and wrinkled, as we can see right here on this photo. Alright? So, that was for Rorthia area. Stereomonocytogenes is widespread in the environment and has been recovered from soil, water, vegetation, and animal products such as raw milk, cheese, poultry, and processed meats. It also has been known to cause illness in many species of wild and domestic animals, including sheep, cattle, swine, horses, dogs, cats, rodents, rabbits, fishes. And it can be isolated from both human and animal asymptomatic carriers. Okay? So napaka-wide range po no ng mga species na kayang i-infect ng Listeria monocytogenes. Alright? So again, this is classified as a zoonotic pathogen because it could infect both humans and animals. Alright? So this is a non-spore forming organism that could grow in up to 10% sodium chloride. Okay? Kaya niya pong mag-survive sa ganun kataas na amount ng salt. Okay? The optimum growth temperature for Listeria monocytogenes is 30 to 35 degrees Celsius. But because Listeria monocytogenes could also grow at 4 degrees Celsius. A technique called the cold enrichment method may be used to isolate the organism from polymicrobial clinical specimens. Okay. So let's say, for example, you collected a sample directly from the patient or from any other clinical sample that was submitted to you. And then after culturing, you were able to observe that there were multiple different bacteria in one colony. So that means that your isolate or your sample was polymicrobial. Okay. How do we differentiate Listeria monocytogenes from the others? Kapag in-incubate nyo po yung sample ninyo, the polymicrobial, at 35 to 37 degrees Celsius, lahat sila tutubo, di ba? But if you move your culture and incubate it at only 4 degrees Celsius, ang 4 degrees Celsius po ay masyado ng malamig para sa maraming bacteria. So this is no longer optimum for them. But for Listeria monocytogenes, it could continue to grow at 4 degrees Celsius. Okay? So kahit na polymicrobial sila dun sa sample nyo, isa lang ang tutubo dahil mag-isa niya lang na kayang mag-grow at 4 degrees Celsius. All right, so that's for the cold enrichment technique. In wet mount preparations, Listeria monocytogenes exhibits the tumbling motility or end over end motility when viewed microscopically, as shown here in this GIF. Okay, so this is because they have a peritricous flagella. So just a review, kapag sinabi nating peritricous, yung flagella, nasan ito located? It's all over, 'di ba? The organism is completely covered in flagella kapag sinabi natin na peritricous ang kanyang flagella. All right? So ayan po. The picture on the lower left is a scanning electron micrograph of one Listeria cell. So dito po ang ginamit ay electron microscope. Okay? For this one right here. Pero itong nasa taas, ito po, this is a wet mount preparation. So doon natin na-observe no, yung tumbling motility or yung end-to-end motility ng Listeria. Okay? The virulence factors of Listeria monocytogenes includes hemolysin, which is specifically called listeriolysin O. It also has catalase, superoxide dismutase, phospholipase C, and a surface protein. Okay, so anong function nitong dalawang naka-highlight na to? The P60 surface protein induces phagocytosis. Okay, sinasadya pa niya, ini-encourage pa niya yung phagocytosis. Meanwhile, ang listeriolysin O naman po, it is hemolytic and cytotoxic. It could damage those or it could damage those phagocytes, or it could kill macrophages. All right? So ulitin natin, listeriolysin O is a cytotoxic virulence factor that could kill macrophages. Studies have indicated that the ingestion of contaminated food with subsequent spread through the intestine is the primary mode of transmission for listeriosis. Basically, you would ingest the bacteria and then it gets absorbed through the gastrointestinal tract and then it spreads to everywhere else. Okay, so in pregnant women, listeriosis is most commonly seen during the third trimester. Listeria monocytogenes is responsible for spontaneous abortion and stillborn neonates. So, in Filipino, still spontaneous abortion is yung makukunan. Okay? And kapag sinabi naman pong stillborn neonates in stillbirth, this is the death of a baby before or during delivery. So, ipapanganak niya, pero patay na po yung baby. Alright? So, that's for spontaneous abortion and stillbirth. Okay? There are two forms of neonatal listeriosis. Okay, so yung una nating describe, that's maternal listeriosis, but we also have what is known as neonatal listeriosis. So early onset and late onset. So early onset listeriosis results from an intrauterine infection that can cause illness at or shortly after birth. So the result is most often sepsis. But when it comes to late onset, no, ito yung kailangan nating tandaan, mas matagal pa bago natin ma-observe yung clinical manifestations. Okay? Pag early onset, shortly after birth lang, na-observe na natin na may something wrong, di ba? But in late onset, it's going to take 2 weeks after birth, all right? Late onset disease occurs maybe several days to 2 weeks after birth. The affected infants generally are full term and healthy at birth, but eventually they develop meningitis. Invasive listeriosis most commonly occurs in persons who are immunocompromised. Okay. And also, it may occur in older adults or yung mga senior citizens, and particularly in patients receiving chemotherapy. So, for this one, the most common common manifestation of invasive listeriosis is a CNS infection and also endocarditis. Listeria monocytogenes grows well on SBA and chocolate agar, as well as on nutrient agars and broth, such as the brain heart infusion agar or the thioglycolate broth. So, bale, hindi naman po fastidious ang listeria. The colonies are small, round, smooth, and translucent. They are surrounded by a narrow zone of beta hemolysis, which may be visualized only if the colonies are removed, okay? Sobrang liit lang daw po no nung beta hemolysis sa paligid ng Listeria monocytogenes that sometimes you may need to like remove the colonies to see the beta hemolysis underneath that. Okay? Now, in wet ground preparations, Listeria monocytogenes exhibits the tumbling motility or end-to-end motility, which we've talked about earlier. Next, in the motility medium naman po, on the motility medium or SIM, what does SIM stand for? Sulfide indole motility medium. All right. So if we were to use the SIM, we would be able to observe the umbrella pattern or inverted Christmas tree pattern. Okay. So in this example, dito sa photo here on the left, this is what it looks like if you were able to observe the umbrella motility of Listeria monocytogenes. Okay. Pero ayun po no, in some references, medyo mas kilala yung inverted Christmas tree. All right? But anyway, ayan po, that's for the motility of Listeria monocytogenes on the sulfide indole motility medium. All right? So this is usually observed if you incubate this organism at 22 to 25 degrees Celsius. Okay, ulitin po natin. The umbrella motility of Listeria monocytogenes is usually observed when incubated at room temperature, approximately 22 to 25 degrees Celsius. Listeria monocytogenes could also produce a positive camp reaction. Okay, so kaya rin po natin gamitan ng camp test ang Listeria monocytogenes at positive din dito ang Listeria. It's capable of producing a block type hemolysis, which would produce somewhat a rectangular pattern. Okay, so ayan po, no? As we can see right here in this example. So yan po, yung block type hemolysis ng Listeria monocytogenes, 'di ba? So for other organisms, they appear to be arrowhead or bow tie in shape, no? But for Listeria monocytogenes, it is block type. So parehas din po yung principle, no? You take Staphylococcus aureus which produces, anong pinoproduce ng Staph aureus? Beta lysine, 'di ba? So, Staph aureus would produce beta lysine, Listeria monocytogenes would produce the camp factor. So, camp factor plus beta lysine equals enhanced hemolysis. So, ayun po, ang pinagkaiba lang ay yung shape. Let's now look into other differential tests. First is the hippurate hydrolysis test. So this detects the production of hippurate and could hydrolyze hippuric acid. Okay, so ayun po, no, if the bacteria produces the enzyme hippuricase, okay, Listeria monocytogenes, it would produce the enzyme hippuricase, it could hydrolyze the hippuric acid to glycine and benzoic acid. Okay, ulitin po natin yung explanation na yon. On the test media, mayroong hippuric, or merong hippuric acid. Okay, on the test media, we have hippuric acid. But if we have Listeria monocytogenes could produce Hipuriate. So hipuriate would hydrolyze hippuric acid. What is the end product of this? Benzoic acid and glycine. All right. Ngayon, yung glycine po na na-produce through that reaction, this can be detected after the addition of ninhydrin, forming a purple colored product as you can see right here in this photo. Okay? So ulitin po natin. Listeria monocytogenes produces hippuriate, which is an enzyme. This enzyme can break down or hydrolyze hippuric acid. When hippuric acid has been hydrolyzed, it forms anong end products? Benzoic acid and glycine. Now, glycine is colorless. We can't see that. So we have to add ninhydrin. All right. So glycine plus ninhydrin equals a purple color. And a positive result is produced by Listeria monocytogenes. Okay. So if ever there may be any confusion between Listeria monocytogenes versus Corynebacterium species, you could perform hippurate hydrolysis because Listeria monocytogenes will test positive for hippurate hydrolysis. Up next, we also have bile esculin hydrolysis. Does anybody remember which organism did we first talk about bile esculin hydrolysis? It's the Enterococcus species, di ba? Go back on our discussion on the Enterococcus species. Pwede natin silang gamitan ng bile esculin hydrolysis. So if a bacteria is capable of growing in the presence of increased salt concentration plus 4% bile and, no, tatlo yung criteria. We they're also capable of hydrolyzing esculin to esculitin, then we would be able to observe a positive result. Okay, so ulitin po natin yung tatlong bagay na kailangan nating ma-observe sa bile esculin hydrolysis: survive the presence of increased salt concentration, survive the presence of 4% bile, and hydrolyze esculin to produce esculitin. Okay? So yung esculitin po, again, this is colorless. We don't see this right away. So it needs to react with the ferric ions present in the culture media, producing a dark brown or black precipitates and dark brown colonies, as we can see right here in these examples. So again, positive dito ang Listeria monocytogenes. Let's now proceed with the non-spore forming, non-branching, catalase negative bacilli, which would include Erysiplothrix rhizopathiae, Arcanobacterium hemolyticum, Truparella species, and Gardnerella vaginalis. There are three species in the genus Erysiplothrix: Erysiplothrix rhizopathiae, Tonsillarium, and Inopinnata. So, Erysiplothrix rhizopathiae is the only species in the genus that is known to cause disease in humans. It's a gram-positive, catalase negative, non-spore forming pleomorphic rod. Okay, so when we say pleomorphic rod, it's actually a bacilli. All right, but sometimes no, it may show us different cell morphologies, right? So human cases typically result from occupational exposure. Individuals whose work involves handling fish and animal products are at most risk. The usual route of infection is through cuts or scratches on the skin, right? So Erysiplothrix rhizopathiae produces three types of disease in humans. First is erysipeloid. So erysipeloid is a localized skin disease, as we can see right here in this example. So kapag po merong OID sa dulo ng isang word, no, it means that it's only similar to something. Okay? So ang erysipeloid ay, does anybody remember what disease is it similar to? Erysipelas, 'di ba? Which is caused by the Streptococcus pyogenes. All right. So notice, 'di ba? Ang clinical manifestations ng erysipelas is that there is redness, swelling, and there is an advancing border. All right. Which is very similar for this one, 'di ba? There is redness, swelling, and notice, no, you may be able to notice na eventually advance or mag-grow po yung borders nila to spread the infection. All right. So that is erysipeloid. All right. Erysipelosis rhizopathiae could also cause septicemia, which is often associated with endocarditis, and it could also lead to a generalized diffuse cutaneous infection. So, erysipeloid is the most common infection caused by Erysipelotrix rhizopathiae in humans, which is a localized skin infection that resembles streptococcal erysipelas. Okay, so that's what I was saying earlier. No, it's very similar to erysipelas caused by Streptococcus pyogenes. So the lesions usually are seen on the hands or fingers because the organisms are usually inoculated through work activities or yun nga po, occupational exposure. The incubation period is 2 to 7 days, and the infected area is painful and swollen, as we've seen in that photo. On gelatin stab or motility medium, erysipelotrix would produce a test tube brush growth, as we can see right here in this photo. Okay, so ayan po yung tinatawag na parang test tube brush motility or test tube brush pattern, which is very accurate. No? It does look like a test tube brush. So again, we may be able to observe that on a gelatin stab or on a motility medium. Primary human pathogen of the genus Arcanobacterium is Arcanobacterium hemolyticum, which is formerly Corynebacterium hemolyticum. So, nalipat po siya ng genus. Okay? So, pharyngitis that is caused by Arcanobacterium hemolyticum can be mild or severe and is often clinically indistinguishable from pharyngitis that is caused by beta hemolytic streptococci. Okay, magkahawig lang daw po, very similar ang strep throat and ang pharyngitis na dinudulot ng Arcanobacterium hemolyticum. Most patients develop cervical lymphadenopathy, so nag-enlarge daw po yung lymph nodes dito sa parang neck area ng pasyente, and approximately 50% of patients develop a pruritic scarlatiniform rash and desquamation on the hands and feet. So parang nagkaka-rashes daw po dun sa hands and feet ng pasyente. Right? Arcanobacterium hemolyticum produces small colonies on sheep blood agar that demonstrates a zone of beta hemolysis after 24 to 48 hours of incubation. Again, very similar in appearance to the beta hemolytic streptococci. Frequently, a black dot is observed on the agar when the colony is scraped away. So, ayun po, no, pitting of the agar. Okay, pitting of the agar is seen beneath the colony, and it also has been reported. So basically, kapag daw po tinanggal mo yung mga colonies or you scrape away some of the colonies either by black na tuldok sa ilalim or, ayun po, when we say pitting the agar, parang nagkaroon ng maliit na butas dun sa agar where the colony was above. Alright? So, ayan po. Next, we can also perform CAMP tests on Arcanobacterium hemolyticum, but on CAMP tests, it exhibits a reverse CAMP reaction, otherwise referred to as CAMP inhibition reaction, because the hemolysis produced by the beta lysine producing Staphylococcus aureus is inhibited by the phospholipase D that is excreted by Arcanobacterium hemolyticum. All right? So, ayan po. They are, or as we can see right here, no, kung ikukumpara po natin, we have right here, let me just use my pointer. We have right here yung Staphylococcus aureus, di ba? Ulitin natin, anong pinoproduce ng Staph aureus? Beta lysine. All right. We have right here the group B streptococci or U streptococcus agalactiae. So again, group B strep produces the CAMP factor. So CAMP factor plus beta lysine equals enhanced hemolysis, 'di ba po? So the positive result for group B strep is the bow tie or arrowhead appearance, 'di ba po? That's a positive CAMP reaction. Pero for Arcanobacterium hemolyticum, babalik ta rin niya. Imbis na mag-enhance yung hemolysis, lalo pa yung ininhibit yung hemolysis. And again, this is because of phospholipase D. Okay? Arcanobacterium hemolyticum produces phospholipase D, leading to a reverse CAMP reaction, as we can see right here. So instead of having an enhanced hemolysis, mas lalong na-prevent or na-lessen yung hemolysis na na-observe natin. Okay, so that's for the reverse CAMP reaction of Arcanobacterium hemolyticum. Next is the true Corella species. In 2011, some members of Arcanobacterium were moved to Truparella. So right here we have two, yung Truparella pyogenes and Truparella bernardii. Ang kailangan lang po natin tandaan dito is anong mga types of infections natin sila pwedeng ma-observe. So if you would notice, both pyogenes and bernardii could be isolated from wound infections. Okay? But for Truparella pyogenes, this has been documented to be transmitted through contact with cattle. All right? So maybe we could say that zoonosis or zoonotic din po yung transmission ng Truparella pyogenes. Gardnerella vaginalis is a short, pleomorphic, gram-positive rod or coccobacillus that often stains gram-variable or gram-negative. Okay, so ulitin po natin, it has a gram-positive cell wall, but sometimes it may appear gram-variable. So paiba-iba yung gram-staining reaction na pwede nating ma-observe. Alright? So, Gardnerella vaginalis has a gram-positive cell wall. However, the peptidoglycan layer is thinner than those that are found among other gram-positive bacteria. Gardnerella vaginalis is primarily known for its association with bacterial vaginosis in humans. Now, for years, Gardnerella vaginalis was thought to be the only cause of bacterial vaginosis. However, studies have shown that bacterial vaginosis is actually a polymicrobial disease, okay, meaning it is one disease that could be caused by multiple different organisms. Okay, so Gardnerella vaginalis would be included, and other bacteria could also join in, such as Prevotella species, Peptostreptococcus, Porphyromonas, Mobiluncus, and Fusobacterium, and also Mycoplasma hominis and Ureaplasma urolithicum. No? Ang haba ng listahan, marami pala sila nagjo-join together to cause bacterial vaginosis. It's just that in the past, no, people thought mag-isa ni Gardnerella vaginalis na nagdudulot ng bacterial vaginosis. So, bacterial vaginosis is characterized by a malodorous discharge and a vaginal pH greater than 4.5. Bacterial vaginosis generally results from the reduction of the lactobacillus population in the vagina. So, 'di ba, yung lactobacillus species po, no, it has kind of like a commensal relationship with the overall environment in the vaginal tract of females. So, after that, no, kapag bumaba yung population ng lactobacillus, this is followed by an increase in the vaginal pH. So, imbis na slightly acidic, magiging alkaline yung pH, no? Vagina, which leads to the overgrowth of Gardnerella vaginalis and other bacterial vaginosis associated organisms. The medium of choice for Gardnerella vaginalis is the human blood bilayer tween agar. So, when cultured on human blood bilayer tween agar, the colonies are beta hemolytic, small, gray, and opaque, as you can see right here in this photo. Gram staining of vaginal secretions is generally regarded as the reference method for diagnosing bacterial vaginosis. So for this one, we should be able to observe clue cells. So clue cells, these are large squamous epithelial cells with gram-positive and gram-negative bacilli, and also gram-variable bacilli or even cocobacilli. Marami nga sila, di ba? So there's a lot of them clustered on the edges and even on top, as we can see right here on this photo. Okay? So sobrang natatabunan na ng mga magkakaibang bacterial cells yung epithelial cell ng pasyente kapag mayroon silang bacterial vaginosis. Alright? So ayan po. Another test that was done in the past is the whiff test. All right. So as the name implies, no, after the addition of 10% potassium hydroxide, as the name implies, a fishy odor would develop, and the person doing the test would have to take a whiff take a whiff of that mixture. And ayun po, if a fishy odor is observed, then this indicates that the infection is probably bacterial vaginosis. All right. So I safely assume people don't do this anymore kasi may gram staining na nga po. All right. So that was for Gardnerella vaginalis. Let's now proceed to the non-spore forming, branching, Aerobic actinomycetes. Cardia species are aerobic, branched, gram-positive bacilli. Aside from being gram-positive, Nocardia species are also partially acid-fast, meaning they're able to retain the primary stain only when a weak acid is used as a decolorizer during the acid-fast staining process. Another term um, for this description would be modified acid-fast positive. All right, ulitin po natin. Nocardia species, although they are classified as gram-positive bacilli, we could also observe them through acid-fast staining because they are partially acid-fast. So you could perform the modified acid-fast staining to visualize Nocardia species microscopically. So the colony and microscopic morphology, as well as the types of infections caused, sometimes resemble those of fungi. All right, so magkahawig daw po yung mga diseases associated with the Nocardia species versus fungal organisms. So you'll talk more about those pagdating niyo po sa Microbiology 2 classes ninyo, where you talk about mycology and virology. Nocardia species can be recovered on standard non-selective media. However, growth may take 1 week or longer. Okay, so hindi naman sila fastidious in a sense that they require specific nutritional sources, but they are slow growing. Okay, incubation period would take 1 week or longer. Colonies of Nocardia species might have a chalky matte or velvety or powdery appearance, and they may be all of this. Ang daming colors na nakalista dito. White, gray, pink, peach, tan, orange, tan, pigment. There's a lot. It may vary. All right. So ayan po. They can have a dry, crumbly appearance similar to breadcrumbs. Kaya yun po yung pinaka-notable na description sa colonies ng Nocardia. They are wrinkled, chalky, and have a breadcrumb appearance. As we can see right here in this third photo. Okay, so yan po yung parang breadcrumb colonies ng Nocardia species. Okay? This first photo right here is what they look like upon gram staining. So again, ito yung dinidescribe na gram-positive bacilli in long, thin, branching filaments. They look like they're connected together, forming these different branches. No, so that's what we mean when you say branching filaments. And then this is what they look like if we were to perform modified acid fast staining. Okay, so parang ganun din po, no? Nag-iba lang yung reagents and yung stain, nag-iba lang yung color nila, pero ganun pa rin po, they are long branching filaments. All right? So ayan po. Generally, infections caused by nocardia occur in immunocompromised patients. So, opportunistic po ang nocardia species. The most commonly encountered species are Nocardia brasiliensis and Nocardia cereosigorgica. Okay, so Nocardia brasiliensis causes actinomycotic mycetoma, which is a form of subcutaneous skin infection, and Nocardia cereosigorgica could cause bronchopneumonia. Okay, so ulitin po natin. For Nocardia brasiliensis, ang target niya ay yung skin and subcutaneous tissues. But for Nocardia cereosigorgica, it targets the respiratory system. Let's now look into the other actinomycetes. Aerobic actinomycetes of clinical importance belonging to the genus Actinomadura include Actinomadura majore and Actinomadura peleateri. They cause mycetomas which are identical to the mycetoma that could be caused by nocardia. The microscopic morphology and colony morphology of Actinomadura species are very similar to Nocardia species. So magkamukha sila sa napakaraming aspects. All right? The organisms can be differentiated using metabolic variations. So Actinomadura majore is celiobiose and xylose positive. Okay? So pwede kang mag-carbohydrate utilization test gamit yung celiobiose and xylose. All right? So positive dito, positive for carbohydrate utilization ng cellobiose and xylose ang Actinomadura majore, but Nocardia species do not produce acids from these two carbohydrates. Next is streptomyces. Streptomyces species are primarily saprophytes found as soil inhabitants, and they resemble other aerobic actinomycetes with regard to morphology and the disease that they cause. Okay, so magkahawig lang din daw po yung appearance and diseases na dinudulot ng streptomyces. Next is Gordonia. Members of the Gordonia are aerobic, catalase positive, gram positive, or gram variable partially acid fast bacilli that are non-motile. Okay, so they are distinguished from nocardia by their ability to reduce nitrate in the absence of mycelia. Okay, ulitin po natin yon. Nitrate reduction positive ang Gordonia and hindi nagbabranching filaments ang Gordonia. Okay? So Gordonia species are typically isolated from environmental sources, and so far reports of human infections have been rare. In almost all cases, patients were immunocompromised as a result of underlying diseases, and infections by Gordonia species um, occurred only secondarily. Right? So in this example, we have Gordonia bronchialis. Up next, we have Rhodococcus equi. This is the most common human isolate in the genus Rhodococcus, and this is found in soil and could cause respiratory tract infections in animals. Human infections is rare, although an increased incidence in immunocompromised patients has been observed. So, opportunistic lang din po ang Rhodococcus equi. Contact with farm animals and feces is an important risk factor. So lung infections account for about 80% of human diseases. Now, on gram staining, Rhodococcus equi may demonstrate filaments with some branching. Okay, Rhodococcus equi may be partially acid fast or acid fast. Now, the key features of identification of Rhodococcus species is its salmon pink pigment, as we can see right here. So, ito yung pinakailangan nating tandaan. Kapag nakita po natin that this bacterial organism is a gram positive bacilli that is mildly branching, and nakita natin na yung appearance ng colonies niya has a salmon pink color, then that would indicate that your isolate is Rhodococcus equi. Okay, keep that in mind, ha? Salmon pink colonies, Rhodococcus equi. Next is Tropharema. Tropharema whipple is the agent of the Whipple disease. Tropharema whipple has been detected in human feces, saliva, and gastric secretions, and is apparently ubiquitous in the environment, so it's everywhere. If untreated, this is a uniformly fatal disease with typical symptoms of diarrhea, weight loss, malabsorption, arthralgia, and abdominal pain. Eventually, neurologic and sensory changes often occur with the Whipple disease. Diagnosis is best made by microscopic examination and also endoscopic biopsy specimens. So that was for all the other actinomycetes. Let's now proceed to Bacillus species. Members of the genus Bacillus stain gram positive or gram variable, pero mas kilala po sila as gram positive. They are aerobic or facultative anaerobic bacilli that could form endospores. Okay, so ito na po, spore forming po ang Bacillus species. The survival of Bacillus species in nature is aided by the formation of spores, which are resistant to conditions to which vegetative cells are intolerant. Okay, so again, endospores, which are produced by Bacillus species, are highly resistant structures. They are capable of surviving harsh conditions. Okay, they're very difficult to eliminate. All right, so ayan po. All Bacillus species are motile with their peritrichous flagella except for Bacillus anthracis and Bacillus microides. Okay, ulitin po natin. Bacillus species are motile. Anong type of flagella? Peritrichus. Maliban sa Bacillus anthracis and Bacillus microides. Upon gram staining and microscopic examination, they are large and very rectangular. So that's why they have been described as boxcar shape. Okay, so I did a quick search, no, kung ano ba ang boxcar. All right, these are actually shipping containers. All right, so ayan po. Of course, they also have their characteristic unstained spores. Okay, so yung mga endospores po that were produced by Bacillus species, they are unable to kind of like have a stain. Okay, hindi po sila na-stain. So as we can see right here in this example for Bacillus species, this is what it looks like kapag nasa middle yung endospore nila. Okay. Virulence factor of Bacillus anthracis depends on a glutamic acid capsule and three component protein exotoxin. The capsule, which protects the organism from phagocytosis, is a polypeptide of D-glutamic acid. All right. So marami na tayo na pag-usapan na organisms with capsules in the previous chapters, diba? So what you need to remember is their specific component. So in cases of Bacillus anthracis, their capsule has a or is composed of D-glutamic acid. All right. Recall time. Recall time muna po tayo if the capsules of Bacillus anthracis are composed of glutamic acid. Ano naman po ang composition ng capsules ng Streptococcus agalactiae? It's sialic acid, as we can see right here in this pop-up screenshot. Okay, so ulitin po natin. Tatandaan nyo po yung mga organisms na may capsules and kung may special component yung capsules nila. So again, for Streptococcus agalactiae, its capsule is composed of sialic acid, but for Bacillus anthracis, its capsule is composed of D-glutamic acid. Meanwhile, the anthrax toxin consists of three proteins. All right. So the protective antigen PA, EF, and LF, each of which is individually non-toxic, but together they act synergistically to produce damaging effects. Okay, ulitin po natin, yung virulence factor po ng Bacillus anthracis is the anthrax toxin. May tatlong components yung anthrax toxin na yon, yung protective antigen, yung EF at saka LF. Okay? Separately, they are non-toxic, but if they work together, they are responsible for the diseases and damaging effects that we will be talking about in the next slide. As mentioned earlier, they are facultative anaerobes, so they could also grow in acidic environments. They're also halophilic, so mabubuhay po sila with up to 7% salt concentration. And nabanggit ko na po kanina, no, that they are motile maliban sa Bacillus anthracis, 'di ba? So non-motile ang Bacillus anthracis, siya lang yung naiiba, 'di ba? All other Bacillus species are motile maliban sa Bacillus anthracis. May isa pa, ano yung isa pa? Bacillus Mycoides. All right. So both Bacillus anthracis and Mycoides are non-motile. Everybody else is motile because they have peritrichous flagella. Now, this bacteria is recognized as a Category A bioterrorism agent by the Centers for Disease Control and Prevention. Okay, so there are three categories of bioterrorism agents. And ang Category A po, this is the highest priority among them. All right. So this include organisms that pose a risk to national security because they could be easily transmitted from person to person. They result in high mortality rates and have a potential to cause major public health impact. All right. They could also cause public panic and social disruptions, and they require special action for public health preparedness. Okay. So those are the things that we need to look out for to be able to kind of like decide, no, kung paano nagiging category A yung mga bacteria na inaaral natin. Let's now talk about the four clinical forms of anthrax. Humans are infected primarily as a result of accidental or occupational exposure to animals or animal products. Cutaneous anthrax begins as a small pimple or papule that appears at the site of inoculation 2 to 3 days after exposure. So a ring of vesicle would develop, and the vesicles would coalesce to form an erythematous ring. After that, a small dark area would appear on the center of the ring, and eventually it ulcerates and dries, forming a depressed black necrotic central area, which is known as the black scar, as shown here on this first photo. So this black scar is normally 1 to 3 centimeters in diameter, but sometimes they could be more extensive and would begin to heal only after 1 to 2 weeks. Okay. Next is inhalational anthrax, also referred to as the wool sorter's disease. So kaya siya tinawag na wool sorter's disease because ulitin natin, Bacillus species, they are soil inhabitants, 'di ba? So in countries where they take care of sheep, ano kinakain ng sheep? Grass, 'di ba? So the sheep are just outside. They feed from the grass and the soil. And when they do that, sometimes yung mga bacterial cells na nasa grass, they get dislodged and eventually kumakapit ito dun sa wool nila. Okay? So kaya tinawag na wool sorter's disease, 'di ba? In countries where they take care of sheep, kasama na rin po dito na parang um harvest, is harvest the right word? They kind of like harvest the wool, 'di ba? They shear the wool to make like coats, hats, and other garments. So kapag nangyari po 'yun, nadidislodge naman yung mga bacteria na kumapit dun sa wool ng sheep papunta dun sa person who's doing the shearing. So nai-inhale nila 'yon. Kaya siya tinawag na wool sorter's disease. So mode of transmission is via inhalation, and this resembles upper respiratory tract infections where there is mild fever, fatigue, malaise, dyspnea. So napaka non-specific nung symptoms, 'di ba? So ayan po. Just in case there are people here who don't know, no, dyspnea means there is shortness of breath or difficulty breathing. Hindi ko na ilagay dito pero kasama rin po sa listahan ng um clinical manifestations ang cyanosis. Okay, so cyanosis, this is the bluish discoloration of the skin due to lack of oxygen in the blood. Next, this could also lead to pleural effusion. So pleural effusion is the buildup of excess fluid in the layers of the pleura outside the lungs. Okay, in simpler terms, may tubig sa paligid ng baga ng pasyente. All of those would eventually progress to disorientation, coma, and death. Okay, so yun yung problema sa pulmonary anthrax. It starts off as something that's non-specific, no? It only resembles upper respiratory tract infections, which could be caused by a lot of things, diba? But eventually, magpo-progress yung infection, and there is now fluid buildup in the lungs, there is disorientation, coma, and death. All right, so that's for the pulmonary anthrax. Up next is gastrointestinal anthrax. This occurs when the spores of the inoculated or spores are inoculated onto a lesion in the intestinal mucosa after the ingestion of the spores. Okay, so yung spores daw po ng bacteria, yung na-ingest nung pasyente. The symptoms of gastrointestinal anthrax include abdominal pain, nausea, anorexia, and vomiting, and bloody diarrhea could also occur. Okay, so that's for gastrointestinal anthrax. Up next is injectional anthrax. All right. So this has emerged more recently and is recognized as an additional form of clinical infection. The infection results from direct injection of spores into tissues, generally during the administration of drugs of abuse. Okay? So ayun po, no, ulitin po natin. Injectional anthrax is common among IV drug abusers. How does that happen? Di ba usually for IV drug users, they don't really practice the septic technique. They don't, for us who are in the medical field, alam po natin na yung mga syringes and needles, single use lang, 'di ba? Pagkatapos nito, dispose na. For IV drug users, it's not like that. Okay? They use one needle and hanggat hindi pa to nasisira, paulit-ulit nitong gagamitan. Ang problema po dito, minsan, they don't cover it back up using the cap nung needle. So, ang mangyayari, iiwan lang nila dyan. And of course, ulitin natin, Bacillus anthracis is present in the environment, 'di ba? Lalo na in the outdoor environment, soil, whatever. So sometimes the bacterial spores present in the outside environment contaminates the needles. All right. And when the needles are contaminated, they inject that onto themselves, leading to injectional anthrax. So the clinical manifestation for this one is walang black scar, but there is a soft tissue infection. All right. So yun po, those are the things that you need to remember for the 4 forms of anthrax. Ano yung mode of transmission nila, and ano yung prominent clinical manifestation nila. So again, for cutaneous anthrax, we usually observe this through or acquire this through cuts and abrasions on the skin. And the prominent clinical manifestation is black scar. Next, for pulmonary anthrax, mode of transmission, inhalation. Clinical manifestations, magsisimula siyang non-specific, matatapos siya sa coma and death. All right? And there's also pleural effusion. Next is gastrointestinal. Ang mode of transmission ay ingestion. Diba? Prominent clinical manifestation, bloody diarrhea. Next, for injectional anthrax, anong mode of transmission? IV drug abuse. Anong clinical manifestation? Walang black scar, but there is soft tissue infection. So those are the four forms of anthrax. Proceeding with laboratory diagnosis, if the clinical sample is suspected to contain Bacillus anthracis, it is strictly advised that we process this in a BSL-3 laboratory. Okay, biosafety level 3 dapat po ang processing ng Bacillus anthracis. So, nabanggit ko na po kanina na yung itsura nila after gram staining. They are gram-positive bacilli that are very rectangular in shape and they have central spores. Okay? Yung endospores nila nasa gitna ng bacterial cell. Okay? We could also perform the Schaefer-Fulton endospore staining as we can see right here. So, ang primary stain po ng Schaefer-Fulton is malachite green, which would stain the endospores green. And then the counter stain for this one is saffronin, which is why the rest of the cells would appear red. For the colony morphology of Bacillus anthracis on BAP or SBA, they are non-hemolytic, large, gray, and flat with an irregular margin because of the outgrowths of long filamentous projections. All right, so because of this, the term medusa head has been used to describe the colony morphology of Bacillus anthracis. So as you can see, just like the medusa head, merong isang solid na area sa gitna. And then on the outside, no, yung mga hair snake things dun sa head niya, they're the filaments, okay? Medyo mas filamentous or mas manipis yung growth sa paligid, all right? Kaya siya tinawag na medusa head. Take note that they are non-hemolytic. Okay, so ito naman po yung itsura nila on cap. So medyo similar pa rin naman, filamentous pa rin naman po sila. Next, the egg yolk agar is used to determine if Bacillus anthracis has the ability to produce lecithinase. So if they do, an opaque zone of precipitate will be produced in the immediate surrounding of the colony, as we can see right here. So again, dito po ang ginamit ay egg yolk agar. So we would like to find out if the isolate produces the enzyme lecithinase. And again, a positive result is indicated by a precipitation surrounding the colonies. All right. So again, you use the egg yolk agar for lecithinase testing. All right. Next is the PEA or phenyl ethyl alcohol agar. This is recommended for the identification of Bacillus anthracis from fecal specimens. Also, the PLET medium or the polymyxin lysozyme EDTA thallus acetate medium, pwede rin po itong gamitin on contaminated specimens aside from stool, which is why, and this is what their colonies would look like. Okay, dito sa fourth photo that we have. Next, on gelatin stab, although they are non-motile, they could produce a growth pattern described as the inverted Christmas tree, as we can see right here. Okay, so ito po yung inverted Christmas tree pattern ng Bacillus anthracis. All right, recall time. Recall time po. Which other organism is capable of producing the inverted Christmas tree pattern? It's Listeria monocytogenes, di ba po? So, ayun po. Next, anthracis is generally susceptible to penicillin, specifically at 10 units per ml. Okay, so ulitin natin, kung 10 units per ml ng penicillin ang mayroon, hindi mag-grow ang Bacillus anthracis dahil susceptible nga sila. But if we could grow them in a culture media with only about 0.5 units of penicillin, so that's significantly less, di ba? Sobrang konti lang. We may be able to observe the enlargement of their cells, and this would in turn change their colony morphology, giving them the string of pearls appearance. As we can see right here. Okay? So dito po, no, ang goal ay papatubuin natin yung Bacillus anthracis sa isang media kung saan may konting penicillin enough na hindi sila mamamatay, but enough na magbabago yung itsura nila. So again, this is penicillin susceptibility. As you can see right here, nabawasan yung pagiging filamentous nila at medyo mas naging shiny sila. Okay? So again, that is referred to as the string of pearls reaction. Bacillus aureus is a relatively common cause of food poisoning and opportunistic infections in susceptible hosts. Yung food poisoning po, just a side note, kahit po hindi kayo immunocompromised, you may still be affected. Food poisoning caused by Bacillus aureus takes two forms, diarrheal and emetic. The diarrheal syndrome usually associated with ingestion of meat or poultry, vegetables, and pasta is characterized by an incubation period of 8 to 16 hours. Okay, so ano pong nangyayari dito, no? Kaya tinawag na fried rice bacillus, yung Bacillus cereus. Madalas na observe ito in take-out food or fried rice from restaurants. Tapos, they leave it out in the open. Okay? So, imagine, nag-take out ka ng fried rice, tapos hindi mo naubos, tapos iniwan mo lang dyan. And then when you come back to it and try to eat it, dun na po maaaring mag-develop yung food poisoning. Why is that? Ulitin natin, saan galing yung Bacillus species? They are soil inhabitants, diba? They're present in the outside environment. So although it's on the inside of your house, diba? Hindi naman natin may iwasan that spores, bacterial spores could be everywhere, all right? So if you leave your food, your fried rice, whatever, pasta uncovered, no, after some time, maaaring makontaminate ito ng Bacillus cereus that's present in the environment, okay? So when that happens, this could lead to, ayan nga po, food poisoning. So its primary virulence factor includes motility, penicillin resistance, and the production of heat-stable enterotoxins. When we say heat-stable, kahit daw po initin nyo yung pagkain nyo, hindi masisira ang structure ng mga enterotoxins na to. So tuloy pa rin yung progression ng disease. Okay? So in diarrheal type of food poisoning, affected individuals have abdominal pain, diarrhea, and vomiting. All right. So, multiple episodes of diarrhea in a day. No, fever is uncommon, but fever may also be present in some patients. All right. So that's for the diarrheal type of food poisoning. Next is the emetic form. So emetic form has the predominant symptoms of nausea and vomiting 1 to 5 hours after ingestion of the contaminated food. Diarrhea may be present in about one-third of the affected individuals, pero mas less commonly observed yung pagkakaroon ng diarrhea. So this form of emetic food poisoning has been associated particularly with the ingestion of fried rice that has been prepared in some Asian restaurants. Okay? And ayun nga po, no, previous history lang yun. But applicable din po ito if you have leftover rice or food that has been improperly stored. Okay? So the average duration of the illness is 9 hours. So for both the diarrheal and emetic forms of Bacillus cereus food poisoning, the illness is usually mild and self-limiting. Okay? So ano kailangan nating tandaan dito? Paano natin sila ma-differentiate? For the diarrheal type, mas matagal yung incubation period, 'di ba? 8 to 16 hours. Tapos ang predominant clinical manifestation, diarrhea. Mas madalas na nagda-diarrhea yung pasyente, hindi masyado sa vomiting. But for the emetic type, baliktad, maigsi lang yung incubation period. In the next 5 hours or less, may simptomas na agad. And anong predominant clinical manifestation? Vomiting. It's less likely na nagda-diarrhea yung pasyente. Okay? So I hope you understood how to differentiate diarrheal and emetic type of food poisoning. So again, this is specifically caused by Bacillus cereus, the fried rice bacillus. Culture of suspected food from a food poisoning incident may be done to isolate and quantify Bacillus cereus. Okay, so dito po, our specimen of choice is a food sample, hindi yung clinical specimen from the patient. Okay, we would take a sample from the food item that you suspect was the causative agent of the infection. Bacillus cereus could be grown aerobically at 37 degrees Celsius on BAP or SBA. Now, on BAP, they would produce beta hemolytic frosted glass appearing colonies, as you can see right here in this photo. Okay, so I hope you're able to tell the difference no between Bacillus cereus versus Bacillus anthracis. Ang Bacillus anthracis, non-hemolytic and filamentous. Okay? Pero ang Bacillus cereus, beta hemolytic and ayan po, frosted glass appearance. Hindi siya filamentous. Okay? So ayan po. Um, all right. So most food poisoning cases no caused by Bacillus cereus do not require antimicrobial treatment. Kasi ulitin nga po natin, self-limiting lang naman yung mga infections na dinudulot nito. This GIF right here shows us a time lapse of the growth of Bacillus cereus. Well, dinagdag ko lang, mahilig kasi ako sa GIF, di ba? Notice there are so many moving images in my presentations. All right, so that was for Bacillus cereus. Listed here are a few other Bacillus species, which we will no longer be discussing in severe detail. So, first up, we have right here Bacillus subtilis, Bacillus licheniformis. We also have right here Bacillus circulans, which is now known as Nealia circulans. All right, Nealia circulans. Next is Bacillus pumilus and Bacillus sphaericus, which is now known as Lysinibacillus sphaericus. Okay, so take note of those bacillus species na nagbago na po yung pangalan. Okay, so Bacillus circulans is now known as Nealia circulans. Meanwhile, Bacillus sphaericus is now known as Lysinibacillus sphaericus. Listed here are diseases associated, so food poisoning, pneumonia, meningitis, bacteremia, basically the same um, types of infections na kayang idulot ng iba pang bacillus. Okay? So, commonly, these are just observed as contaminants. Dahil ulitin natin, bacillus species are present in the environment. Here are the references that were used in the making of this discussion. That ends our discussion on Chapter 16 of Mahon's textbook of Diagnostic Microbiology. This has been Aurel Enriquez. Thank you so much for watching.